Carrageenan is a material that can be obtained from red seaweeds, which is cultured and harvested in ocean farms in the Philippines, Indonesia, and Malaysia. This material is used as a binder in processed meat, providing a material texture and consistency for keeping its volume intact in storage. Its water retention properties keep the processed meat juicy and not dried or burned when cooked or baked. The property to gel at room temperature extend the applications in other products as a stabilizer and emulsifying agent in dairy products, beverages, and juices, and as a thickener for creams and sauces. To provide more information, let us hear from Dr. Romel G. Bacabag.
3 types of carrageenan are commonly used by industry. Kappa, the rigid version of this material, iota, the soft version, and lastly, the lambda, which does not form a gel. In our previous study, mixtures of kappa and iota carrageenan exhibited an effective averaging effect in the final properties of the end product. Carrageenan mixtures preserve the properties of the iota and kappa components, resulting into a viscoelastic hydrogel. Carrageenan mixtures have predictable properties that can be associated with the concentration of the iota and kappa components, where an increase in kappa concentration makes the mixture more rigid. To exploit the natural biocompatibility of carrageenan, we explore its potential in the fast-evolving 3D printing technology. We have recently established the MBG Center for Tissue Engineering and Biological Soft Materials as a niche center for Region 7 under the Science for Change program of the Department of Science and Technology. Through this new research center, we will develop novel and relevant biomedical applications in collaboration with the Materials Development Laboratory of the Advanced Manufacturing Center of DOST, in partnership with the MCPI Corporation of Consolacion Cebu, with expert advice from the Orthopedic International Incorporated and from our scientific collaborators in the Acta Vrije Universiteit Amsterdam and the Tokyo University of Marine Science and Technology. In the 3D-BTM project, we will develop 3D printable and injectable scaffolds using carrageenan mixtures for controlled final properties to biomanufacture functional artificial bone, muscle, and pancreatic tissue constructs. Through our bioinks, we envision bringing 3D printing technologies to bridge the gap between medical imaging and surgery through effective collaborations between experts and surgeons in the service of the Filipino patient. Daghang salamat!